Okay, so this is our next step. So we've gone through transcription where we've created a pre-mRNA, okay? And that was our whole process, our last lecture on creating the pre-mRNA. Then we went through modifying the pre-mRNA by adding the cap and the tail, okay? And then removing the introns, and now we've got an mRNA. And the mRNA now is in the cytoplasm. So the next step is now we're going to take that mRNA and we're going to translate it into protein. And that's going to be our lecture on translation. Okay, so let's just make this one clear. So here is our code. Now this would be mRNA. This is not pre-mRNA, and this is the kind of sequence we're going to go through. mRNA is going to be fed through a ribosome, and these codes are going to be broken down into triplets or codons, and every 3 bases is going to code for an amino acid. So here's an amino acid, here's a tRNA, and we're just going to basically keep adding and making a polypeptide, which is a protein. Okay, and this is a complex process. So we're going to be talking about today's translation from mRNA to protein. We already have an mRNA. We're going to turn it into protein. Okay, and the idea is how a code of nucleotides becomes a code of amino acids. So here's some good links. Okay, there's lots of really, really good links up there. If you're having trouble finding some, you can use mine. But really just looking into Google and looking under translation, I mean, you really do want to watch videos of this. Okay, it really, really helps out. Okay, so translation overview. The whole purpose is to translate the mRNA code into an amino acid sequence to make a protein. So here's our mRNA code. We wanted to eventually get down to this, which is a series of amino acids or a protein. Okay? It happens in the cytoplasm, and there's a couple big players here. We got mRNA, ribosomes, tRNA, which are called translation factors. Okay, so here is a picture of a tRNA with a little bit more detail than we normally have. So tRNA, or transfer RNA, is an RNA molecule in a cloverleaf shape. So we got a kind of a cloverleaf shape. This is how all RNA molecules look. Okay, there are 2 main functional units of a tRNA. The anticodon loop, and that's right here, which I have kind of X'd here. Okay, and this is where the most important part is. It's where the anticodon is. Okay, so the anticodon is going to be complementary to the codon on the mRNA. Okay, and this little code here is going to tell basically the ribosome as to which tRNA or which amino acid is going to be next. Okay, a couple things I want you to add on here is the mRNA gets read from 3' to 5'. So you want to add that to your notes, that mRNA gets read from 3' to 5'. Okay? And the tRNA is going to be in the opposite direction. It's going to be 3' to 5' in its orientation. So mRNA is going to be going this way. So it's going to be heading to the right. So it starts reading on this side, and then it's going to keep going and keep going. The opposite, it's going to be the anti-parallel 3' to 5'. Okay. So besides the bottom part here, the only other part that I need you to know is the acceptor stem, and this is where a specific amino acid for that tRNA attaches, and that's just before the anticodon. Okay. So this is what's going to happen. So codons are located on the mRNA, and they are written in a 5' to 3' direction. So here's my codon, okay, and mRNA is being read to the right. So it's going to start with the 3' end, and then going towards the 5' end. So it reads the 3' end first, and then to the 5'. Okay, the opposite is going to be on the anticodon. It's going to be 3' to 5'. Okay, so codons are located on the mRNA are written in a 5' to 3' direction. Okay, but it's read 3' to 5', and the anticodons on the tRNA are written in the 3' to 5' direction. Now, since each amino acid contains its own anticodon or tRNA, there are 20 different tRNAs. Okay? Because each tRNA contains its own unique amino acid based on its anticodon sequence. Okay, so now let's get to the last player, which is the ribosomes. So the ribosomes, okay, as well as rRNA, okay, are used as a meeting place for mRNA and tRNA. So that's our third RNA, which is rRNA, and that's just a ribosome. And there's 2 main parts of the ribosome. We got the large unit, and that's going to be this big part here with the EP and A attached. We call that the large subunit, and then the bottom is the small subunit. Okay? And on the large subunit, we got 3 binding spots, and we'll talk about that in a second. So, first thing, translation is very energy consuming. It takes a lot of energy to do this. According to our second law of thermodynamics, anytime you take simple molecules and make them into complex, you need to have energy, and we're definitely doing that here. We're taking, simple amino acids and making them into a polypeptide chain. So that's going to require energy to create order. So translation involves a ribosome facilitating the union between an mRNA molecule, use our mRNA molecule, and the tRNA codons. It's basically kind of a meeting spot for both of them. Now translation, just like DNA transcription and replication, occurs in 3 phases: initiation, elongation, and termination. Okay? Here's the whole process. Now the key here, and I want you to make sure that you pay attention, okay, is the ribosome is going to keep sliding to the right. The mRNA does not move at all. Okay? The mRNA is stuck. It's not going to be moving at all. But this ribosome is going to keep sliding down here. Okay? So it's just going to keep sliding down through the mRNA molecule. Okay? So this keeps sliding. So this is going to slide down. And then when it slides down, these next 3 bases are going to be in the A spot. Okay? Because as the ribosome moves down a little bit, it's going to slide this A spot over to UGC, and that's going to be the next amino acid sequence that's going to be added to this. Okay. So let's slow down a little bit and go through it in phases. So let's talk about the transition initiation phase. So during initiation phase, all components come together. The small ribosomal subunit, the large ribosomal subunit, the mRNA, and the initiator DNA. First thing, step 1, the small ribosomal subunit attaches to the mRNA near the start codon. So we don't have the large subunit attached yet, just the small subunit. And then we have our first amino acid, and like it always should be, it's methionine. So it's going to be AUG. Okay? So we've got methionine. That's my first amino acid sequence. It's the start of all of the proteins. Okay? That's our first step. Okay? So we have the small ribosomal subunit attaches to the mRNA. So we got mRNA and the small subunit. Step 2, the initiator tRNA anticodon, UAC, attaches to the mRNA. Okay? It attaches to the mRNA. And all eukaryotic, the initiator tRNA is carrying a methionine. It's going to start with methionine. Okay? So that's step 2. Step 3, the large ribosomal subunit attaches to complete the ribosome. Okay? Notice that methionine is already there when the large subunit attaches. Okay? And when that large subunit attaches, notice that methionine is in the P spot. Okay? It's in the P spot, not the A or not the E, but in the P spot. Okay? That's important. For the methionine, the first amino acid joins to the P spot. Okay. So my very first one is going to be P. Now every other tRNA that comes in is going to first join to A, then it's going to slide to P, then it's going to slide to E. Okay, it always goes in this order, A, P, E, except for the methionine, which is just going to go P, E, and then out. Okay. So what happens here is we have basically our first amino acid was here. Our next tRNA comes and bonds to the A site. Okay? What's going to happen is this whole polypeptide chain is now going to break off the tRNA that's in the P site, okay? And it's going to come join onto the A site. That's going to leave this tRNA empty, okay? Because it's lost its whole entire chain. It's going to attach here. This tRNA is going to be empty. It's going to slide over to the E site and then release. Okay? Once this tRNA that's in the A site gets this long polypeptide chain attached to it, it's going to then slide to the P site and everything slides down. Okay? So looking at it here, we got their A, P, and E site. So what's going to happen, the next step is valine is going to disconnect from this tRNA here, and it's going to come and join onto this lysine. I got my arrow there to kind of indicate that. That leaves this tRNA empty. It's going to, this one from the E site is going to kick off. This P is going to slide over to the E and then release, and the A's are going to slide to the P. Okay, and then we're going to keep going to the sequence. This one, next one's going to now join into the A spot. Okay, so let's go through the elongation sites. tRNA with methionine is in the P spot. That's where we start. The next tRNA and the appropriate amino acid will bond to the A site. So we had our first one, methionine. It was in the P site. Okay, all these other ones all came in just like this one did. It's going to come into the A site. So we come into the A site. Okay, this long polypeptide chain is going to jump off, join onto the A site. Okay, this tRNA is then going to slide over to the exit site and then leave. Okay, and that's what we have here. This one, this P, okay, transferred its polypeptide chain over the A site. This one's then going to slide over here and then kick off. Okay, the one that the tRNA that's in the A site. See how this one's going to come and join here? Now we have this long polypeptide chain in the A site. Well, then it's going to slide to the P site. The P site's going to be vacant because this one has slid over to the E site. And then we just keep going and going and going. We keep adding polypeptides until we're all done. Okay? And the process just keeps repeating itself. So finally, when a stop codon is read, so we're going to keep making this polypeptide chain until we get a stop codon. When the stop codon is read, everything disassembles. The large subunit, the small subunit, okay? And then we just have this free polypeptide, which is a protein now. And that protein can go on to do whatever it needs to be done. Okay? This last thing, this is just showing you the amino acid sequence for insulin. So this is what insulin is. Notice that my first amino acid in insulin is methionine. Okay? And then here's all of the amino acids in order for insulin. If you had one of these amino acids out of order, so say right here where I have a proline, say this proline somehow got switched and it was glycine instead of proline, that would make this protein inactive and you would be diabetic. If you're diabetic, you have a mutation in your DNA. And that means that you can't create the proper mRNA sequence, which means you can't create the proper mRNA sequence, which means you can't create the proper amino acid sequence, and therefore you can't create a proper protein. Okay. So I hope that kind of clarified things on protein synthesis. Now the big thing here is that you got to go watch videos on it. Okay? I can do all of the explanation that I want in terms of text and kind of still pictures, but you really do want to watch an animation. I mean, it's by far the best way of learning about protein synthesis is by watching these videos. And I've kind of already asked you to do that, and most of you have already done that assignment and emailed me. And why I did that is because, you have to really go outside the classroom sometimes because these topics get way too complex. Okay? And anything in science is going to be like that. There's going to be lectures that you would get in science, especially in maybe chemistry, if you're going to take chemistry, and physics and biology, where you watch a lecture and you're like, what the heck just happened? I have no clue as to what the heck they were talking about. Well, you can't just forget it, right? You can't just be like, oh, well, forget it. You have to learn that material. So if the professor did a poor job of explaining it, or say you just had have a poor understanding of it, maybe they did a great job, but you just can't understand it, make sure that you go through extra resources to figure that out by watching YouTube videos, by looking at worksheets. Okay, I thought it was excellent, some of the people that have already emailed me. You beginning to find your own worksheets. And I don't want you to think that, oh, Mr. Bunning's being a lazy teacher, making us find our own worksheets. I'm kind of preparing you for university. That's what my goal is. I don't want to be lazy by any means. What I'm trying to do is show you how you prepare. Okay, and those are the kind of things that I didn't learn until third or fourth year. Well, I want you to know it in first year. So when you walk into university, you're one of these super students that says, oh, I didn't get lecture, I'm going to YouTube and I'm going to find some YouTube videos on it. Or, oh, I didn't like the YouTube videos, I'm going to find a worksheet to ensure that I actually understand it. Because once you are a complete independent learner, and that's what I'm trying to make you as an independent learner, then it doesn't matter how good of a professor you have. You're going to do well in the course regardless. Because you're going to have good teachers, you're going to have bad teachers, you're going to have good professors, you're going to have bad professors. And you don't want your mark to hinder on the performance of your teacher or professor, especially in university when you may get some very, very bad professors. Okay, like ones that I had were horrible professors, but I still need to get the information. So if I can be an independent learner, which I'm trying to make you, then, irregardless, sorry, that's not even a word, regardless of the professor or teacher in front of you, you're going to be able to get that information because you can teach yourself. Okay? So hopefully this was helpful. Email me with any questions about protein synthesis. Okay? I have a worksheet there that I would have liked you to work on as well in the news feed.